Welcome to this explainer. Okay, I want you to imagine a clinical sample for a second. Whether that's a swab, some pus, milk, or feces, don't just think of it as a medical specimen. I want you to picture it as a chaotic, microscopic crime scene. Because down at the cellular level, it is absolutely packed with different microorganisms, all just living right on top of one another. And to figure out exactly what's causing a problem, we really need to investigate this scene. We have to separate the innocent bystanders from the actual culprit. So today, we're diving into the fascinating process of how we do exactly that, the isolation of microorganisms from clinical specimens. Here's the roadmap for our little investigation today. First, the quest for pure cultures. Then, tools of the trade. Third, the streak plate method. Next, reading the microbial clues. And finally, why isolation matters. Let's kick things off with Section 1, the quest for pure cultures. Right, so whenever a specimen actually comes into the lab, it is essentially this messy microscopic soup. You've got pathogens hiding right there among the totally harmless normal flora. And just like in real life, you can't interrogate a whole crowd at once to find out who committed a crime, right? You have to separate your prime suspect from the rest of the group. That is the entire principle of bacterial isolation, moving from that chaotic, mixed population of bacteria to a highly organized, completely separated single type of microorganism. So let's lock in exactly what we're aiming for here. A pure culture is strictly defined as a culture containing only one type of microorganism. Isolation is just the physical process we use to pull that pure culture out of the mixed population, because once we finally have a pure culture, we've got our isolated suspect ready for questioning. That brings us to Section 2, tools of the trade. Well, how do we physically separate these tiny microbes? You're going to need some very specific gear. Petri plates to hold your media, and obviously a marker to label everything, because trust me, you don't want to mix these up. But the absolute star of the show here is the inoculating loop. That's your primary vehicle for moving our microscopic suspects around. You also absolutely need a heat source, like a Bunsen burner or a spirit lamp, just to sterilize that loop. Plus, an incubator set to exactly 37 degrees Celsius to help the bacteria grow once we've separated them. And depending on what kind of bacteria we're looking for, we actually use different types of culture media to grow them. Nutrient agar is your standard general-purpose medium. But if you have fastidious bacteria, which is just a fancy way of saying they're kind of picky eaters and eat really rich nutrients, you'd use blood agar. Then we have what we call selective media. These are special environments that only let certain suspects grow. For example, McConkey agar is specifically for gram-negative bacteria, and mannitol salt agar is what you'd grab if you're trying to isolate Staphylococcus. Okay, so we've got our wire loop, our agar plates, our incubator, but here is the real puzzle. How do we actually use these tools to separate millions of completely invisible microbes from one another? How do we make the invisible visible? Let's answer that in Section 3, the streak plate method.
Now, while there are other techniques out there, like the spread or port plate methods, the streak plate is pretty much the gold standard for isolating pure colonies. The whole process builds across 5 really meticulous steps. Step 1, labeling your plate. Name, date, specimen type. Crucial. Step 2, sterilize your loop in a flame until it is literally red hot, and then let it cool. Step 3, grab a small amount of the specimen. Now, step 4 is where the magic actually happens, the streaking. You streak the specimen across the agar in 4 progressive quadrants. What you're doing is physically diluting the bacteria across the surface so much that individual, totally separated colonies will eventually form from single bacterial cells. Finally, step 5, incubate that plate inverted at 37 degrees Celsius for 24 hours.
Of course, a quick reality check here. When you are handling clinical specimens, strict biosafety and aseptic precautions are absolutely non-negotiable. You always need to work near the flame and avoid opening the Petri plate unnecessarily. You really want to prevent any air contamination. Always flame your loop before and after you use it. Double-check your labeling, wear your lab coat and gloves, disinfect your work area, and wash your hands. If you skip any of these, you risk contaminating your pure culture, or honestly worse, exposing yourself to some nasty pathogens. Moving right along to Section 4, reading the microbial clues. So, fast forward 24 hours in the incubator, and our suspects finally reveal themselves. We can actually identify our microscopic culprits by observing their colony morphology. That just means looking at their physical characteristics as they grow on the agar. We're looking at a bunch of features here.
The overall size of the colony, its shape, is it a perfect circle or is it all irregular? Its color, its surface texture, and its elevation. Like, is it flat against the jelly or is it convex and raised up? And these visual traits, they practically act like confessions. Different bacteria literally give away their identity through these striking clues. For instance, if you see golden yellow colonies, that very often points straight to Staphylococcus. E. coli famously shows up as these bright pink colonies when you grow it on McConkey agar. Bacillus colonies tend to be really large and rough-looking. And Pseudomonas totally gives itself away by producing this really distinct green pigment. Just by observing these traits from our pure culture, we are well on our way to making a positive ID.
Finally, let's wrap up with Section 5, Why Isolation Matters. So, why do we go through all this meticulous streaking and observing? Well, because isolation isn't just some abstract lab exercise. It has massive real-world stakes. It's totally essential for identifying bacteria and for running antibiotic sensitivity tests, which is how doctors know exactly which medicine is going to cure a patient. Taking veterinary microbiology as an example, this exact process is used every single day to diagnose really severe animal diseases. We're talking mastitis, hemorrhagic septicemia, salmonellosis, brucellosis, and some seriously nasty wound infections. Without getting that pure culture first, an accurate diagnosis and treatment would be pretty much impossible.
It's honestly amazing when you think about it. Just a simple piece of wire and some agar in a little plastic dish literally hold the power to sort out the microscopic world, diagnose severe diseases, and ultimately save lives. By mastering the very methodical steps of the streak plate method, we can unlock the secrets hidden within any clinical specimen. So I'll leave you with this thought. With millions of undiscovered microbes still out there, what other invisible culprits are just waiting for the right streak plate to finally be unmasked? Keep exploring, keep learning, and I'll catch you in the next explainer.